The title of my poster is Evidence of a Plausible Epigenetic Effect on Equine Cell Differentiation under Heat Stress. It has been well established that extreme weather events such as storms, flooding, fires, and so on, or extremes of temperature, high or low, can cause stress on the body and mind. As an example, Project Ice Storm at McGill University under Suzanne King studied a cohort from the 1998 ice storm in Quebec of pregnant mothers, and they showed epigenetic effects occurring during development that continued into childhood development. Epigenetics, or from the epigenome, are the factors that affect the phenotype, the observable outcome, outside of the genome, that is, the gene sequence. The epigenome can be altered based on the environment, but it may also be inherited, and this is called the developmental origins of disease hypothesis, or Barker hypothesis. Existing research, however, is based on small populations with data that's difficult to reproduce, and looks at cohort studies of individuals in a very variable population. To understand the molecular and cellular mechanisms of this stress, we needed a cellular model of development under stress. This led us to our research question: what is the impact of stress on the proteome and cell differentiation of an equine stem cell model? What we did is use primary mesenchymal stem cells, or MSCs, taken from horses at the Atlantic Veterinary College at the University of Prince Edward Island, and differentiate these stem cells into osteoblasts, bone-forming cells, and adipocytes, or fat cells, and compared with cells of the same type, differentiating at 42 degrees instead of 37 degrees to act as a heat stress. We then took a look at the proteomic profiles derived using mass spectrometry using tandem mass tags so that we could compare relative protein levels in different samples. As you can see from the poster, what we ended up with were a series of proteins that changed in differentiating cells under stress that we colored based on whether they were increased or decreased in relative abundance in the stressed cells. Red for if they were increased in the stress cells compared to the non-stressed control cells, or blue if they were in fact lower in the stress cells. And we also grouped them based on protein function and similarity, or protein in the same protein families. And these are in the green and black lines that connect the circles, as you can see from the poster. As it can be observed, there is little overlap between the pattern between the osteoblast and adipocyte results, suggesting two different reactions to stress in differentiating cells, therefore demonstrating that the type of differentiation was responsible for the reaction to the stress. In the case of the osteoblasts, the majority of proteins were increased in the stress cells, whereas the adipocytes mostly showed decreased protein abundance in the stressed cells. Interestingly, for the adipocytes, one of the few proteins, two in fact, that were higher in the stressed cells was vimentin. Vimentin is an intermediate filament, a component of the cytoskeleton, that is involved in differentiation under stress in neurons, among other functions. However, vimentin has also been shown to inhibit osteoblast differentiation, which makes a lot of sense as we found it to be higher in the adipocytes under stress and lower in the osteoblasts under stress, suggesting a role in inhibiting osteoblast differentiation in adipogenic differentiation, and the reduction in the level of vimentin having the reverse effect in the osteoblast differentiation. Overall, we found proteins that were involved in signaling pathways, in cell signaling, such as the Wnt and Notch signaling pathways, proteins involved in the structure or morphology of the differentiating cells, like cytoskeletal proteins, as well as proteins involved in endocytosis. For future work, the first step would be to find whether these were in fact epigenetic or genetic effects, whether these were causing mutations on the genome or epigenome levels, whether these same effects would be observed in human cells compared to equine cells, as well as other stressors. We simply used heat as the most simple and easy to control stressor. However, these results could be expanded to other stressors, such as chemical stress, inflammatory stress, and so on. Thank you for listening to my presentation.